Hello guys, welcome to the DMS online school. On this channel, I teach mathematics, biology, science, and English, and also revise past examination papers. So in today's video, I'm just going to decide to revise this paper that was written on the 11th of November 2021. This paper reads Examinations Council of Zambia, Examination for School Certificate Ordinary Level, Biology, Paper 2, Theory. This was written on Thursday. All right. So without further ado, let's just get started. All right. So this is question number 1, and question reads: Figure 1.1 and Figure 1.2 show freshly cut potato chips suspended in solution E and F at the beginning of an experiment and after 2 hours. Okay, so we have two figures here. Figure 1.1 at the beginning and then figure 1.2 after 2 hours. So in figure 1.1 we have two solutions, solution E and solution F. In solution A we have freshly cut potato chips put in solution E here, and also the same freshly cut potato chips put in solution F. Then after 2 hours we notice that potato chip in solution E has this shape, and then potato chip in solution F has this shape. So in solution E, at the beginning, this was the shape. Then after 2 hours, this is the shape. Okay. In solution F, this is the shape of the potato chip, and this is the shape of the potato chip after 2 hours. Then A part 1 says explain the difference in the appearance of the potato chips in solutions E and F after 2 hours. Yeah. So guys, I'm just going to be reading the answers very fast, and I'm not going to explain very much because the explanation of these lessons happen in my online revision. So this is just a revision so that you see how we do it in our online revisions, how simple, how easy we put our answers, very clear and straightforward. So when they say explain the difference in appearance of the potato chips in solution E and the F after 2 hours, we are going to say that the difference in the appearance of the potato chips in solution E and F after 2 hours is that the potato chips in solution E are shrunken and less fresh, while the potato chips in solution F are swollen and firm. Yeah. So here they've shrunken. The potato chip in solution E has a shrunken or shrinked, and the potato chip in solution actually F has actually swollen and become firm. Yeah. So let's proceed. Then part 2 says, based on the appearance of the potato chips in solution E after 2 hours, what term could be used to describe solution E? So we need to see the term that can be used to describe solution E here. All right. So the term that can be used to describe solution E is the term to describe solution E is hypertonic. This means that solution E has a higher concentration of solutes than the potato cells. Then part B part 1 says what would be the state of cells in the potato chips in solution F after 2 hours. So what would be the state of cells in the potato chips in solution F after 2 hours? So we are in solution F here. So what will be the state of cells in potato chip in solution F after 2 hours? So here, guys, we must understand that the state of cells in solution F after 2 hours is turgid. All right. Then we are saying this means that the cells have absorbed water and become swollen and firm. Okay. Then part 2 says state the importance of the state of cells in B part 1 above to plants. Yeah. So like they're asking about the importance of turgidity in cells of plants. So we are saying this is important for plants because it helps them maintain their shape and structure. So turgidity is very important to plants. Why? Because it helps them maintain their shape and structure. Then C says, explain the meaning of the term isotonic solution. Simple. So the term isotonic solution means a solution that has the same concentration of solutes as the cells. In an isotonic solution, there is no net movement of water across the cell membrane. All right, so let's proceed to question 2a. Table 2 shows various food tests carried out by a learner. So we have in this column here nutrients, in this column test, in this column observation, in this column conclusion. So here we are told the nutrients is starch, the test is left blank, the observation color change from brown to blue black, then conclusion starch is present. So now if they're telling you that you have a nutrient as starch, then color change from blue to black, what is the test? It is iodine. All right, so here we call it iodine. Then here, nutrient, we don't know. Test is emulsion, then observation, white precipitate, conclusion. So if you have emulsion, the emulsion is the test that is used to test fat. So the nutrient here is a fat. Then if it produces white precipitate, then that means that fat is present. Yeah. Then protein, then test, nutrient is protein, test is burate, then observation dash, then conclusion, protein is present. So if protein is present when you test using burate, then the color should have been purple. Yeah. Then this is actually, we have answered for part 1, which says complete the table by filling in the spaces. So we have done it. Then part 2, which nutrient in the table would cause kwashiorkor if lacking in the human body? So if among these nutrients, the nutrient that would cause kwashiorkor if it is lacking in the human body is actually protein. Okay. Then part 3 says name a nutrient not in the table which helps to prevent constipation. So the nutrient which is not in this table but helps to prevent constipation is actually a roughage fiber. Then here should have been, this is part 3, then this should have been B. State 2 deficiency diseases in plants. So number 1 is the iron chlorolysis. All right, so it is iron chlorolysis. This is a condition where the leaves of plants turn yellow due to lack of iron. Then the number 2 one is a blossom end rot. This is a disorder that affects the fruit of some plants due to calcium deficiency. All right, so if I'm going fast, don't worry, because if you join my online lessons, I actually explain these things. So this is a quite quick revision, but it can help you to understand how to answer these questions in biology. So if you're enjoying this video, please consider liking it and let me know in the comment section. And if you haven't yet subscribed to the channel, please consider doing so by clicking on the red subscribe button, so that when I post prediction videos, you won't be able to miss. Those interested in the online lesson, the line is going to be left on the screen here. All right, so let's proceed. So we have question 3, which says figure 3 shows a longitudinal section through a root tip. So this is a root tip and this is a longitudinal cross section. We have letter here. Okay, here we have vesicular bundle. Here we have a G, this part. Then this part is labeled H. Then this part is I. Then this part is J. Then M says identify the parts labeled G and J. So what is G here? So G is root hair. Okay, as you can see from this one here. So when you look at this, I just brought this one. When you look at this one, this is the part here. So we have vesicular bundle, root hair, this one here. This one is called apical meristem. This one is called root cap. Then this region from here up to here is called area of division. This region is called area of elongation. Then this one is called area of maturation. All right, so let's go back to the question now. When they say identify the part labeled G and J, so this one we say it is going to be root hair, then this J is going to be root cap. Then part B says which labeled part in figure 3 is responsible for generating new cells. So the one which is responsible for generating new cells is actually I here. So I is responsible for generating new cells, and which is called the apical meristem. All right, so let's proceed. Then this one here says region H is responsible for cell division. All right, then part 1 says apart from vesicular bundles, state two other cell types differentiated by region H. So we need to see which region H are we talking about here. So this is region H. So we are told according to the question that apart from vesicular bundle, state two other cell types differentiated by region H. So the other cell type is cambium cells and also parenchy, parenchyma cells. Yeah, this is quite tricky to pronounce. Then part 2 says state two types of cells found in a vesicular bundle. So which vesicular bundle are they talking about? This one here. Which two types of cells found here? So the types of cells found in vesicular bundle are number one, xylem cells, and then number two, phloem cells. Then part 3 says which named type of cells in C part 2 shows movement of substances upwards and downwards depending on the needs of the plant. So like the ones we have mentioned here, xylem cells and phloem cells, which of these two allows movement of substance upwards and downwards depending on the needs of the plant? So it's actually phloem here. That's the answer. Okay. So we proceed to D. Suggest one meristematic. Suggest one meristematic structure not in the diagram responsible for bringing about secondary growth in dicotyledonous plants. Should be vesicular cambium. Okay. So the answer is vesicular cambium. Vesicular cambium is a meriste. Okay, meristematic structure that is responsible for bringing about secondary growth in dicotyledonous plants. All right, so let's proceed to question 4. Figure 4 shows the structure of a nephron. So this is a structure of a nephron. This part is labeled K. This part is labeled D. This part L here should have been labeled. Actually, it is labeled L. Yeah. So then they say identify the parts labeled A, identify the parts labeled K and L. Okay, so the part labeled K here is the afferent asterial, and then the part labeled all right, sorry. So here I saw that maybe it was M, so that's why I say proximal covulated, but this one should be the glomerulus. So let me just change quickly the answer. Yeah, so part L is the glomerulus, see, already changed. Then B part 1 says name two substances present in the fluid in K but absent from the fluid in M. So we are talking about the two substances that are present in K here, but they are absent in M here. So blood cells, and then part 2 is proteins. So proteins are the ones that are present in K, but they are not present in M here. Then we proceed. Part 2 says give two reasons for your answer in B1, or in B part 1 above. So why we say that here, why we say that here in K there is protein and then there is also blood cells. Those are the reasons they want us to give answers for. So number one is that blood cells are protein and proteins are part of the blood. Yeah. So they are part of the blood. Okay. They are part of the blood because blood as it comes here, it carries the blood cells, the proteins, all right? So they're present there, they will be there. But the other reason, why are they not here? So this is the reason that we are giving. So the reason is that blood cells and proteins are too large to pass through the glomerular filtrate membrane here. So why we discover that in this part here, we have proteins and blood cells, and yet in M here we don't have them, is because proteins are large as well as the blood cells. They are large, so they cannot pass through the membranes of the glomerula. Because of that, they are not found here, but they are just found in that region. I hope you understand. Then we move. Then C part 1 says state one common disorder associated with the kidney. So this is kidney failure. Then part 2 says explain how the disorder in C part 1 can be treated. We are saying kidney failure is treated by dialysis machine and kidney transplant. Yeah, that's what is used. Then we come to question 5, which says figure 5 shows 4 animal cells at different stages of mitotic division. So yeah, so this is cell 1 at a different stage, cell 2 at different stage, cell 3 at different stage, cell 4 at different stage. Then they ask us to say, A, name the structure, the body, N and the O. So this structure, the body, actually N here, you say this one should be the nucleus, should be the nuclear membrane, sorry. So this layer here is the nuclear membrane. Then this structure here is actually chromosome. If you want, you can call it chromatid. So this one is a chromosome actually. Then we move. Then B says, at which stages of mitotic division are cells 1 and 3? Okay, so like these cells 1 and 3 here, 1 and 3, which stage of mitotic division they are. Okay, so like cell 1 here, we can say that cell 1 is at the anaphase. Okay, then cell 3 is at prophase. Okay, so you have to understand these stages if you are to join my online lessons for biology, where I went in detail in explaining these two processes which are found in cell division. All right, so then part C says, a husband and wife were heterozygous for blood group A and B respectively. Then they're saying, with the help of a genetic diagram, show the possible blood groups of their children. Yeah, so this is about the blood groups. We have actually three types of blood groups. We have blood group A, we have actually four. Yeah, we have blood group A, we have blood group B, then we have blood group AB, then we have blood group actually O. Yeah, so these are blood groups that we have, which are determined by about three alleles, which is A, B, and O. Okay, those are alleles which are there to determine these groups. Now, when we have a husband and a wife that are heterozygous for blood group A and B respectively, what does it mean by heterozygous? So for them to be heterozygous, it means that their alleles, they have got two types of alleles in them. Okay, so the father has got the A group alleles. In this, for the father to have the blood group A, there are two alleles that are determining the blood group A, and then for the mother to have blood group B, there are two alleles. That's what it means by heterogeneous. Now, what you must understand is that let me just get something to explain here quickly. Very like that I'm explaining something here. So here are the blood groups here. So if you have A, then we have blood group B, then blood group O. If A is heterogeneous, okay, meaning that in A there is actually A is dominant. When they say they've got the blood group A and it is heterogeneous, meaning in A there's blood group A, or and also in B there is blood group actually B O. Okay, so sorry, B O. So in this case now, for the father to have blood group A here, it means A is always dominant over B, over O. And for the mother to have blood group B, it means that B is always dominant over blood group over the allele. Yeah, here we have this allele A and this allele O, allele B and allele O. All right. So this one, yeah, we are not talking about that. We are just considering this because of this and that. So if they're heterogeneous and yet the father is said to have blood group A and the mother is said to have blood group B, it means that the father has got AO, but the A is always dominant over O, and then also the mother have got B, and then the B is always dominant over blood group O. Yeah. So now, if they say in this question to say, with the help of a genetic diagram, show the possible blood groups of their children. So here, what we need to come up with first is the parents. So what parents do we have? We have father's blood group, that is crossing with the mother's blood group. Actually, here we have to put the cross like that because this is a crossing. Okay. Now, the next is phenotype. So the phenotype is the physical expression of the gene combination. So in the father, we have seen that in the father, the physical expression of the gene that is expressed is actually A, and then the mother it is actually B. Now, when we go to the genetic makeup, which is called the genotype, in the genes, this is what we find. Instead of having A, we have A O, and also the mother here we have B O. These are the actual genes, but what is expressed physically is A, and here it is a B. Then now we go to gametes. So during fertilization or sexual intercourse and fertilization occurs. These actually split. Okay, this is splits. So the allele A and allele B will split. Okay, and then the allele actually B and allele O splits. So this combination here is in the what we call the uh, 46 chromosomes. Okay, a full individual have got the 46 chromosomes in their cells of the body, but in their sex cells they've got the half of 46 chromosomes, which is 23 chromosomes. So this here says represents what is in the body cells, which is actually 46 chromosomes, but here represents what is in the sex cells, which are gametes, and in the sex cells we have half of what is in the actually 46 chromosomes. So we have a half of A, we have A, and here B. Then here mother also have got a B, I mean A O, and mother B O here. So during sexual intercourse, the father will provide the full set, which is the A and B, but this is how the crossing will come to form the first filial generation of the genotype of the children. So the genotype genetic makeup of the first filial generation of the children will be like this. So this can cross with that from the mother. Okay, so this may have two possibilities. Either this can cross with that and form the combination of AB, or it can cross. This can also cross with that and form the combination of A or there. Then this or can cross with that and form a combination of BO, or it can cross with also that and form a combination of OO. So these are the possible genotypes of the first filial generation of their children. Okay, so this one will have this. Now, the first filial phenotype, what is going to be expressed in their body. So whenever there is a combination of what we call the allele A and allele B, which are the alleles for the type of blood AB, both A and B, when they are combined, they are codominants, which means they are both expressed in an individual in what we call phenotype. So this one, both A and B will occur. But in a situation where we have A and D, or here, only A is dominant. In a situation where we have B and O, only B is dominant. In a situation where we have O, that O, okay, is dominant, and like that. So this is how you're supposed to answer these questions. So let's go to the section B. Now we are done with his section A. Now we are in section B. So here section B says essay question 36 marks. Answer any three questions from this section. All answers must be in complete sentences and paragraphs. Write your answers in the separate answer booklet provided. So you have heard these are what we call instructions, and you need to follow them for you to be able to pass this examination, because they've told you that all answers must be in complete sentences and paragraphs. You need to follow these. So write your answers in the separate answer booklet provided. So there will be different answer booklets that will be provided, and you need only to answer three questions from this section. So let's see how you can answer this. So 6A, they're saying describe the movement of organic solids in phloem. So here the answer says the movement of organic solids in phloem tissue is called translocation. It involves the transport of sugars, amino acids, and other substances from the source where they are produced or stored to the sink where they are used or consumed. Translocation occurs through the sieve tube elements, which are connected by sieve plates. The movement is driven by pressure gradient created by osmosis and active transport. So this is how you should answer this. Read this, you will be able to understand, because the answers are very clear and straightforward. Then we go to part B. Part B, they are saying outline the process of measuring the rate of transpiration using a potmeter. Here we are saying question 6b. A potmeter is a device that can be used to measure the rate of transpiration by measuring the volume of water uptake by a plant. A potmeter consists of a capillary tube connected to a leaf shoot and a water reservoir. The water uptake is measured by recording the time taken for an air bubble in the tube to move a set distance. The rate of transpiration can be calculated by dividing the distance moved by the bubble by the time taken. So this is how you answer this question. You can read this passage here to understand how straight the answers are. Now we move on. This is explain the factors that affect the rate of transpiration. 4 marks. Here we're saying question 6c. We're saying the rate of transpiration is affected by several factors such as temperature, humidity, air movement, and light intensity. These factors affect the rate of evaporation of water from the leaf surface and the concentration gradient of water vapor between the leaf and the atmosphere. Generally, higher temperature, lower humidity, higher uh, air movement, and higher light intensity increase the rate of transpiration. So that is it. You have answered this question. Then we go to 7a. Describe the internal structure of the heart. Then B, describe the factors that reduce immunity to pathogenic diseases. Then C, explain the importance of immunization. So to answer the first question here, we're going to say the internal structure of the heart consists of 4 chambers, which are left ventricle, right ventricle, then we have right auricle and left auricle or atria. It has valves like tricuspid and bicuspid valve. Then the heart is made up. Okay, sorry here. So the heart is made up of the cardiac muscles. So the heart is separated by septum. It has veins which are vena cava and pulmonary vein. So the heart also has arteries like aorta and the pulmonary vein. So this is how you get to answer this first part. Then the next part here describe the factors that reduce immunity to pathogenic diseases. So you're going to say question 7. Factors that reduce immunity to pathogenic diseases include age, sex, genetics, then infection history, stress, malnutrition, chronic diseases. Then here we have immunosuppressive drugs and environmental pollutants. So these are the things that can reduce immunity to pathogenic diseases. Then we move. This says explain the importance of immunization. We are saying, see, immunization is important because it protects individual and communities from serious and preventable diseases such as measles, polio, tetanus, and diphtheria. Immunization also reduces the burden of disease on the healthy system and contributes to global health security by preventing outbreaks and pandemics. Yeah, so this is how you get to answer these questions. You can read and see how clearly the points are. That's how we do it on the DMS online school. So we move. 8 says, A part 8A says, using a named example, describe animal and wind dispersal of seeds and fruits. Then B, explain the importance of fruits and seed dispersal. So we'll start with the first one here because this is 8 marks, so it will be a long passage. So here we are saying question 8M. Animal aided wind dispersal of seeds and fruit is a type of seed dispersal that involves both animals and winds as agents. Some examples are the seeds of the burdock plant have hooks that attach to the fur or feathers of animals. The animals carry the seeds to a new location where they may detach or be removed by grooming. The seeds are then dispersed by wind to a further distance. Then the fruits of the maple tree have wing-like structures that help them to glide in the air. The fruits are often eaten by squirrels who may drop some of them while feeding or store them in their caches. The fruits that are dropped or forgotten may be carried away by wind to a new location. Yeah. Then the fruits, okay, so here you don't need to put these points here. Sorry, I forgot to remove them because we are not writing in points. Then the fruits of the mistletoe plant are sticky and adhere to the beaks of birds. The birds may wipe their beaks on the branches of other trees where the fruits may attach and germinate. The seeds inside the fruits may also be dispersed by wind if they fall off from the fruits. Okay, so we have answered part A of question 8. And then part B says explain the importance of fruit and seed dispersal. Here we're answering to say fruit and seed dispersal is important because it allows plants to spread their offspring to new and favorable environments, whereby where they can germinate and grow with less competition and predation. It also increases the genetic diversity and adaptation of plants by introducing variation and recombination among population. It also reduces the risk of extinction and disease by preventing overcrowding and inbreeding. Yeah. So we have answered question 8. Now we go to question 9. A, which says describe the functions of the following hormones. Part 1, thyroxine. Part 2, glucagon. B, explain the causes of short sight and how it can be prevented. Then C, suggest ways by which deafness could be prevented in humans. So let's start with question part A. Here we are talking of functions. So the functions of thyroxine and glucagon hormones are but one we are saying thyroxine is a hormone produced by the thyroid gland that regulates the metabolism, growth, and the development of the body. It also affects the heart rate, body temperature, and energy levels. Then we come to glucagon. We are saying glucagon is a hormone produced by the pancreas that raises the blood glucose level by stimulating the liver to release stored glucose. It also helps the body make glucose from other sources such as the amino acids. Glucagon is released when the blood glucose level is low or during fasting or exercise or stress. All right, we have answered A, part 1 and part 2. We go to B. B says explain the causes of short sight and how it can be corrected. Here we are saying question 9b, we are saying the causes of short sight and how it can be prevented. Short sight or myopia is a condition where distant objects appear blur while close objects can be seen clearly. It occurs when the eyeball is too long or the cornea is too steeply curved, causing the light rays to focus in front of the retina instead of on it. So we are saying short sight can be corrected by using glasses with a concave lens or laser eye surgery. These methods help to flatten the cornea or shorten the eyeball so that the light rays can focus on the retina and produce a clear image. Yeah, we have answered this one. We got to see suggest ways by which deafness could be prevented in humans. So we are saying question 9c, the ways by which deafness could be prevented in humans are, we are saying number one, deafness or hearing loss can be caused by various factors such as age, genetics, infections, trauma, noise exposure, autoto- autotoxic drugs, and chronic diseases. Some ways to prevent deafness are, so also here we need to avoid points. Sorry, I did forget. So let me just correct this. Yeah, so I've changed. So it's better you number them like you put points because I don't know why biology is just so stingy. They are against this. So we're saying some ways to prevent deafness are number one, getting regular hearing tests and treating any underlying conditions that may affect hearing. Then number two, avoid loud noises and wearing ear protection devices which when exposed to noise environments. Yeah. And then number three, actually here, practice safe listening habits when using headphones or earphones. Then number four should be this one, quitting smoking and avoid exposure to second-hand smoke. Then four, consulting a doctor before taking any medication that may harm here is. Then let's go to question 10a, which says explain the meaning of the following ecological terms. Part 1, habit. Part 2, community. Part 3, niche. Then B, part 1, using named examples, explain what a food chain is. Part 2, explain how energy flows through the food chain. So let's start with the question 10a, habitat, community, and niche. So here we are saying that the meaning of the terms habitat, community, and niche are: habitat, number 1, is the natural environment where an organism lives, such as a forest, a pond, or a desert. Then part 2, community, is a group of different species that live together and interact with each other in a habitat. The niche is the role and position of an organism in its ecosystem, including its food source, behavior, and interaction with other organisms. So here, when you answer like this, you get all the marks under question 10 part A. Then we come to B part 1, using named examples, explain what a food chain is. So here, question 10b part 1, we're saying a food chain is a linear sequence of organisms that eat one another so that energy and nutrients flow from one to the next. For example, a food chain is a grassy in a grassland ecosystem, maybe a glass, then it goes to a rabbit, then it goes to fox. In this food chain, we're saying grass is the producer that makes its own food from sunlight. Rabbit is the primary consumer that eats grass. Fox is the secondary consumer that eats rabbit. Yeah, we go to the other question, but to explain how energy flows through the food chain. Here, question C, we are saying energy flows through the food chain from one tropic level to the next, starting from the producer and ending with the decomposers. At each tropic level, only a fraction of the energy available is transferred to the next level, while the rest is lost as heat, waste, or respiration. The efficiency of energy transfer is typically around 10%, meaning that only 10% of the energy at one level is available to the next level. So this is how you get to answer these questions so that you get all the marks. Now there is a revision package for past paper 2 at going and scripture which can help you to answer these questions. All right, I hope we have come to the end of this lesson, right? Yeah, we have come to the end of this revision. I hope you enjoyed it and it showed you how you can answer biology questions in paper 2. As for now, guys, bye and see you in the next video. Peace.